Okay, this video is part 9 of iron overload lectures. This is iron and the brain. Increased total body storage of iron. As we talked about, men become overloaded when they reach adulthood because they're no longer growing so rapidly. They don't need so much iron. So in their 20s, women tend to become iron overloaded after they stop menstruating, typically in their 50s. Okay, increased free iron means increased oxidative stress. Multiple brain disorders are associated with increased iron accumulation, including the most common vascular dementia, which is mistakenly called Alzheimer's quite often, Parkinson's disease, worse prognosis pretty much in any brain disease with increased iron accumulation, including multiple sclerosis. Okay, here's just a quick couple papers on Parkinson's disease. Increased iron accumulation in the substantia nigra. The substantia nigra also has GLUT4 glucose transporter, so it's sensitive to the same things as diabetes. So here's the papers if you're interested. We're going to go a lot more into the Douglas Kell Etheresia Pretorius theories of iron-related dementia. Oh, there's also papers from, for example, Christopher Exley. He's sort of like the world expert on aluminum. His nickname is Mr. Aluminum. And he believes that aluminum within senile plaques associated with Alzheimer's disease are causing accelerated amounts of ferrous redox cycling of iron. So we talked about that where Fe2 plus cycles back and forth with Fe3 plus, giving off electrons to oxygen in the process and causing hydroxyl radicals, which do a lot of oxidative stress damage to tissues. Okay, so here's all the papers in that. We're not going to get into this too much. Oh, one funny thing though is the guy who's the world expert on chronic cerebral hypoperfusion as a cause of vascular dementia, that's Jack Delatorre, the PhD over here, he says Alzheimer's is not a major cause of dementia, okay? Meanwhile, Exley, who's the aluminum guy, says Alzheimer's is the most important thing. Dr. McDougall also thinks Alzheimer's is the most important thing. Where do I side on it? I think that it's multifactorial. I think aluminum is important for it. I think Delatorre's theory is fantastic, and I also think my theory is quite good. But you see how there's always a little bit of turf involved in all of it. And I'm going to say I think that they all play major roles. Let's leave it at that. And they're all easy to avoid, so you might as well do so. All right. Now, here's sort of the big picture of what's going on. We're going to talk about how iron factors in with dormant bacteria. And this is something that your doctor's not going to know this. Even your AOAs don't know this, top 10% of their medical school class. Only people who really read and study a lot. I told you, I know a whole bunch of infectious disease doctors. I like them. None of them knows this stuff, okay? There's conventional ways of thinking that are in the medical textbooks. This is beyond that. And by the way, there's tons of papers on this. This is some of the most cited papers in the whole literature. Okay, so anyways, what's going on? When you've got leaky gut, you absorb bacteria from your gut into your blood. You also absorb LPS, lipopolysaccharide, which is produced by your gram-negative bacteria. Your gram-positive bacteria produce LTA, lipoteichoic acid. These are both highly toxic, and they do about the same thing, LPS and LTA. All right, so these get into your blood because of the leaky gut. Some of it's cleared by the liver, but some continues on into your blood. Now, you also will have excessive free iron if you're eating a lot of meat, especially red meat, and if you're eating iron-fortified grains and cereals. Okay, all of these things, LPS, LTA, and free iron in the blood, cause an excessive amount of clotting. They make the blood more prone to clot. That's called hypercoagulable is the typical medical word. Now, a normal fibrinogen molecule, that's the precursor protein in the blood for forming clots. Clots are also called fibrin. So when it's activated, the ogen goes off and it just becomes fibrin. Fibrin has hydrogen bonds between different parts of its own molecule. So these are called intramolecular hydrogen bonds. And this shape like a slinky is cylindrical, and that's called an alpha helix shape. So this is one of the standard forms of secondary protein structures to be alpha helix in shape with intramolecular hydrogen bonds. Okay, so what's the point? When contacted by LPS, LTA, or free iron, fibrinogen will distort its shape and it'll become what is called beta-pleated sheets. So beta-pleated sheets are flat like a piece of paper. You can stack them up on each other. You can't stack slinkies on each other. And now what happens is the hydrogen bonds will form in between two different molecules of the fibrinogen. Okay, so now you get stacks of them. So this is an individual fibrinogen molecule. Here are fibrinogen 1, 2, 3, 4 in a beta-pleated sheet secondary protein configuration where they're flat. And they'll then stack up on each other and they'll form big piles. A few of them all stacked together is called an oligomer. When there's a whole bunch of them, it becomes a polymer. And the point, though, is this causes distorted blood clotting and what's called amyloidogenic. So amyloid is when a protein goes from an alpha helix normal cylindrical configuration, like a slinky, to being flat and stacking up on each other. That's called amyloidosis. Okay, this is amyloidosis of the fibrinogen protein. All right, fibrinogen is just a precursor of fibrin. Okay, now the people who really figured this out was Douglas Kell. He's a PhD guy from England, and he was working with Etheresia Pretorius, PhD lady. She's from South Africa, and they really did brilliant research. He's more of a microbiologist. She's more of a hematologist type. And they showed that basically if you got leaky gut or leaky gums, poor dentition, you don't take care of your mouth, bacteria and LPS and LTA are getting into your blood. And they get reactivated when there's increased free iron available. As people progressively become more iron overloaded, they'll have more free iron in their blood. Other things can release that free iron in their blood. If there's liver injury, the dead liver cells will release iron into the blood, for example. And then what happens is you've got dormant bacteria living in your blood that can be reactivated by the presence of free iron becoming available. Once these dormant bacteria are reactivated, they can release LPS. The gram-negative bacteria have LPS. The gram-positive bacteria have LTA into the blood, and that makes it more prone to clot, more prothrombotic. Okay, and if it can get across the blood-brain barrier, which, you know, a lot of people have little strokes, diabetes, and hypertension-related problems with their blood-brain barrier, plus a high-fat diet can increase the blood-brain barrier permeability. And if some of this stuff gets in there, it can cause precipitation of beta amyloid protein in the brain is what's thought to be the mechanism. Okay, here's just some more papers along these lines. You'll find them real easy. You look up Douglas Kell Etheresia Pretorius, you'll be amazed at how many papers they've written and how popular their papers are. Okay, so dormant bacteria being reactivated by iron in the blood, iron overload. This is all the stuff we're talking about. He's got a really nice lecture online, Douglas Kell, and she's got some good lectures online too, but he's got this 1 hour lectures. It's great. Okay. Now, some people say, well, how do you know there's dormant bacteria in the blood? This is well known, okay? We all know you can have TB and sits around for decades till it gets reactivated. We all know you can have syphilis and it sits around for a long time till it gets reactivated. Tertiary syphilis, neurosyphilis. We all know Lyme disease can sit around in the bacteria Borrelia burgdorferi and then be reactivated. And these are just the really famous ones. There's a lot of ones that are in the blood. They're just not that famous, okay? We know that there's weird bacteria that can live inside WBCs like Bartonella. There's also weird bacteria that can, like Francisella tularensis for tularemia, the mycoplasma. So another important dormant bacteria is Micrococcus luteus. So anyways, if you look it up, you'll see that that is well proven. They're much more easy to culture if you culture them anaerobically. In the past, when they would try to culture donated blood aerobically, they wouldn't get very much. But once they did it anaerobically, they found they're far, far, far more common. And actually, the most common one was Propionibacterium acnes, for what that's worth. Okay, so that's an important point. There's plenty of dormant bacteria in the blood. Okay, so by minimizing leaky gut, you minimize those. And by avoiding excessive free iron becoming available to reactivate them, you'll minimize the problems with that. Okay, here's another paper by Kell and Pretorius and another one of their co-workers there. Leaky gut, compromised gut barrier, you get the bacteria, and then they have LPS and LTA, okay? When bacteria are in the wrong spot in the blood, instead of just calling them ectopic, they call them atopo. Same difference. All right, here's an electron microscope showing bacteria in the blood, and here they are showing these abnormal clots. And it will not only cause distortion of the fibrinogen, it also causes distortion of the platelets. This is called blebbing. So it causes a distorted clot formation, and that's bad because these clots are more difficult to lyse. Like we talked about, what happens with free iron, that means normal iron is always bound to something. It's bound to transferrin in the blood. It's bound to ferritin within cells. When it gets free, redox cycling, so Fe for ferrous for iron, and ferrous redox cycling means it's cycling in redox reactions. These are reduction oxidation reactions between Fe3 plus Fe2 plus Fe3 plus Fe2 plus. And in the meanwhile, it's giving off oxygen electrons to these oxygens, and these can form hydroxyl radicals. Fe is for ferrous, and it's also for Fenton, and the Fenton reaction is what this one's called. The Haber-Weiss reaction is also part of all this. Okay, so just to remind you, when you have electron transport in the mitochondria, here's outer mitochondrial membrane, here's IMM, inner mitochondrial membrane, here's electron transport proteins passing the electrons down like a fireman bucket brigade till it gets to oxygen, then oxygen's converted into H2O. Once in a while, an electron will pop off where it's not supposed to. It's called an electron leak, and it'll bind with oxygen in the mitochondrial matrix, but it gets neutralized by SOD, superoxide dismutase. That's good to know. We're going to come back to this enzyme in a later lecture, superoxide dismutase. And then you got other enzymes to help neutralize these free radicals, catalase, glutathione peroxidase. So the body expects this, and it can handle it quite well. But it does not expect free iron to be present in any type of significant amount. And when it is, the free iron predisposes to this Fenton reaction. Hydroxyl radicals can trash these membranes, and it can induce something called lipid peroxidation, a chain reaction of oxidative stress inside the membranes, and destroy these membranes. Okay, so again, we talked about leaky gut, getting gram-negative, gram-positive bacteria into the blood, and them releasing their endotoxins. And everything made worse with the iron, because the iron will reactivate the bacteria. The iron will also distort the shape of the red blood cells and the fibrinogen. So this is predisposing you to these amyloidogenic clots, where instead of having these monomers of fibrinogen with internal hydrogen bonds, you're getting these oligomers and polymers of fibrinogen into fibrin in beta-pleated sheets where the hydrogen bonds are between the two different molecules. And they're stacked up like stacking your hands on each other or a piece of paper. So here's just a couple of the papers. There's a whole bunch of papers on this. Here's a normal red blood cell. It's discoid, the central pallor right in there, a smooth outer surface with excess iron, LTA or LPS. You get distortion of the RBCs like into a teardrop shape, and then you form these distorted matted clots. You get a lot of debris accumulating on the outer surface of the RBC. So here's a normal fibrin clot. It looks like pieces of spaghetti. All right, here it is when there's excess Fe or LPS or LTA. You get these thick matted deposits because it's transitioning into an amyloid form. And that's a big mess. And the relevance is those clots are more difficult to dissolve. The body all the time is forming little tiny clots at bifurcations in your arteries, and it clears them out quite rapidly. But when they're more dense matted deposits like this, it's more difficult for the body to dissolve them, and that's increasing the risk of occluding an artery, causing a stroke, a heart attack, or impotence, for example. Okay, here's just, more papers. These are Kell Pretorius and their co-workers there. You get increased LPS in the blood. LPS binds to plasma proteins like fibrinogen. It distorts the fibrinogen, so it clots abnormally into an amyloid form. And this causes acute hypercoagulation. Okay, so it's creating more work for your body to dissolve these clots. And the body sometimes can't keep up with the clot. Occluded arteries, that's how people die. That's how you kill stuff in the body, because you plug up the artery and you get strokes and heart attacks. Okay, here was a beautiful experiment. Congo red is a standard stain to bind with an amyloid protein. And for this study, they had a Congo blue, a Congo red, and a Congo green sort of variants. We'll call them that. They actually have a little bit different name, these stains, but just think of them that way because that'll make sense. So if amyloid is present, this one's going to turn blue, this one red, this one green. And a normal control clot, you get almost none of this. All right, so what was the point? When you add LPS to the blood, pop, 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 hallelujah, okay? All three stains positive. You add iron to the blood, pop, 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 all three positive, hallelujah, okay? And then LTA from the gram-positive bacteria, same thing, hallelujah, hallelujah, hallelujah. So this is big stuff. What I'm trying to say here is I think Etheresia Pretorius and Douglas Kell deserve Nobel Prizes. I mean, this is big stuff to figure out this mechanism of clotting, this mechanism of amyloid transition of the clot, this mechanism of dormant bacteria reactivation, this mechanism of traversal of the blood-brain barrier with subsequent inducing amyloid protein in the brain to thus itself become amyloidogenic, all right? And then you say to yourself, well, so what if you get amyloidogenic protein in the brain? What's thought to be the mechanism of that? How could a dead protein, even if when something precipitates and you get more and more of them stuck together, the bigger a molecule is, the more likely it is to precipitate out a solution. When it's getting bigger, as it's becoming a polymer, that means more and more components are all added to themselves and stuck together. They can have mass effect. So it's thought that the mass effect of accumulations of beta amyloid protein on the plasma membrane of neurons, like in the hippocampus, a memory center, can push on the NMDA receptor and cause it to open up. And so it'll then allow calcium into the cell. Well, calcium causes activation of the cell, and that'll cause excitotoxicity. If you overactivate a neuron, it will develop what is called excitotoxicity, meaning that it has too much ATP demand relative to what it's able to produce, and that can cause a neuron to die, go into apoptosis. So this is a mechanism by which a mechanical structure can induce cell death of a neuron, apoptosis. In addition, if these are big enough, they can push on the adjacent neurons, which are close by, and you can propagate the abnormality, causing the death of an adjacent neuron. Okay. Plus, when a neuron dies, if it dies rapidly, it'll leak its contents, including its glutamate neurotransmitters, which are excitatory, and it'll overexcite, overactivate the adjacent neurons, and they can die from that. That can cause excitotoxicity, apoptosis in those adjacent neurons. So this is a rather incredible thing to provide this whole, mechanism of causation. And I can tell you, this is very popular in the communities that study neurodegeneration. So here's putting it all together, kind of like the 4th of July grand finale fireworks. Leaky gut and leaky gums from poor dentition, allowing ectopic bacteria to get into the blood. He calls it atopobiosis. All right. And there's also a lot of things that cause leaky gut. We talked about that in other lectures. Now you have established dormant bacteria in your blood. They're often just dormant, like just waiting to be activated by free iron available. Person becomes iron overloaded because they're eating meat and they're eating iron-fortified foods, enriched foods. This causes microbial reactivation. The microbial reactivation leads to release of LPS and LTA, and that'll cause pathological coagulation in amyloid form. This amyloid form can activate beta amyloid, the brain protein, extracellular brain protein, to then also become amyloidogenic and precipitate out of solution and have a bulk mass effect upon plasma membrane and hippocampal neurons. Okay. Then this is the amyloid aggregates of the brain's protein, beta amyloid protein, and those causing neuronal injury in the brain. So anyways, that's a rather incredible thing that Douglas Kell and Etheresia Pretorius figured out themselves. Thought it was very impressive. Okay, so the take-home message is you want to keep your ferritin levels low, probably ideally between 20 to 80, at least below 100. We talked about the excess iron reactivating dormant bacteria. You also want to avoid excess dietary iron, okay? You can consider donating blood. If you don't want to donate a whole unit, donate a half unit. That's what I did, and now I don't even donate anymore. I just pop off a couple tubes. Like if I'm going to go get my cholesterol drawn, I'll draw off a little extra blood. If I'm going to get my vitamin B12 drawn or any other lab I want drawn, check my serum ferritin, I'll draw off a little extra, and I'm gradually getting mine down. I'll present my labs later on, how I got them down. All right, and so all the stuff we talked about in this lecture and other lectures, these are like languages of nutrition, disease causation, pathology, and toxicology. And so all these languages you've learned about carbohydrates, fat, protein, antioxidants, mitochondrial dysfunction, diabetes and insulin resistance, mechanisms of hypertension. It's like speaking in tongues, okay? And here's the El Greco painting of Pentecost. And the little flames represent speaking in tongues. And there's the Holy Spirit right there, the dove. And so here's another great painting of Pentecost, which, by the way, is this Sunday, the 50 days after Easter, the resurrection. And the flame signifies the ability to speak in tongues. Mary in the center is calm. The others are surprised. You now can speak in the language of nutrition and toxicology and improved health. So here's from Matthew 28. Go ye therefore and teach others, in the name of the starch, the fruit, and the vegetable, the healthy Trinity. Teach them to observe all things I have shared with you. And behold, I am with you always, even unto the end of the world. Matthew 28. Okay. So anyways, that's it. Hope that was helpful.